we get experimental science. We're curious, non-judgmental.
Brand new species, and it's already classified as endangered. So let's go ahead and get in talking about this really fascinating newt. These are called crocodile newts. They are not crocodilian in nature. And the new species is really cool for a huge number of reasons. So crocodile newts, there are 39 species of this particular kind of newt. They range from in Asia. They're going from the Himalayas to Nepal, India, Myanmar region of China, Thailand, Vietnam. Why this is a really, really wild, cool study is the location that was identified. Most of these crocodile newts are pretty low in terms of latitude. This one is at an elevation of 1,800 meters. And so this is just the highest level that any this has ever been found. The highest before was 250 meters in elevation. So it's massive on that front. What's really cool and sad about this paper, it's a double-edged sword. They did a population survey, meaning that they did like samplings of the area to identify the density of the animal. So like, are they really populated? Are there lots of this animal? Are there few of this animal? Never been discovered before. Until, well, I believe last week is when the paper actually dropped. Already classified as endangered. This was in Vietnam that this was discovered. So it's already rare now when it was discovered. This is a different species here. Gwen, this is a relative of the one that was identified, but this is the one that was actually in the paper. The researchers tried to hypothesize of why it's already endangered, and their suspicion is because of the coloration. They say that the colors are quite vivid and would be a possible reason for poachers to collect them, unfortunately. So, and I really, like, Gwen, that drives me up the wall where, like, check out this really beautiful animal. Do you know, because it's so beautiful and cute, let's kill it. And so apparently, I think that's the unfortunate, like, approach that the poachers are taking. And, it makes sense insofar as when the researchers are suggesting that this is probably the reason why it's going towards should be classified as endangered, because that seems to be a metric of it. How small? I do have the measurements. This particular one, they say it's moderately sized for a newt, so between 6 and 6.7 centimeters was the range by this particular one. Yay! The paper is, I think at this point, if y'all have been here for the new species studies, it's become very standardized, right? Like every single one of these new papers still pretty much goes along the very similar route. So they did sequencing, they did taxonomy, like to study the size, shape of the animal. They looked at the genetic sequencing and compared sequences between closely related species, looked at the differences between them. One extra thing that they did, which I thought was kind of cool, was they looked at the mitochondrial DNA and sequenced that as well. So they did sequencing on both fronts, which is a nice little bit of something extra. They did sack 6 of them. So 6 animals were utilized for sequencing, as well as this morphological, this shape measurements to figure out how they look and act. But my suspicion is, Smikes, they didn't say this in the paper, but they didn't know that they were endangered until they started doing the collection. And unfortunately, again, new species, but already endangered. Why can't you draw blood for sequencing? You can, Smikes. So what they did was, so it's a double-edged sword, right? Because what we've talked about in the past is if you have one holotype, meaning you have one representative member of that species that you do all your measurements on, how do you know if you're not finding an outlier? Right? So what they did was they got 3 of each. They got 3 male, 3 female, and then took measurements of all 6 and then made average values of them. And then holotypes are usually not let back into the wild, but they're preserved forever. So that if you think that you found a new species and you wanted to compare back to the original holotype, you can go back to the lab or a museum or the university associated with it and look at the holotype. And so they were just doing more, I think, of a due diligence in terms of their science versus just having like the single example that, and which is, we've seen that, Smikes, in the previous weeks of new species, where it's like, here's this brand new animal, and here's just one of them. Yeah, exactly. Yep, like forget the name of the museum at Harvard, but yeah, you can do those comparisons there as well. And they have the pin collection with newts and other animals that can't dry as well as insects. You have them preserved in a jar, usually of formaldehyde or ethanol, but the same principle as well, that you can do your comparative measurements. And I like the fact that they have more than one, because it makes, I think, the data more trustworthy. But again, it's like, well, but they're endangered. But yeah, it's hard to know. These are all the animals that they did sequencing comparisons against. They had 55 different sequencing analyses that they ran. Here is the phylogenetic tree. For those of you interested, that is like a family tree of genetic relatedness, where they're identifying where all these different species are located. So these are all the animals that they analyzed. And here in white are the 2 new species that they identified. So they are able to figure out, that these animals shared a common ancestor with this branch, and they're a different outlier on that front. How many genes do they do all of them? When you do this comparison, so this comparison looks at a global approach and looks for differences on the nucleotide levels. Basically, the metric comes down to is that you have a sequence. You have individual A, and let's say that the sequence is, ATCG, ATCG. And that's at one particular gene. And then you look at individual B, and then they have ATG, ACG. And so then what you have is that you have some gaps in the sequence. There's a gap here, and there's a gap here. Those 2 components are what are analyzed then and compared against. So this is the same gene. This is still, like, let's say this is still gene A. Both of them are gene A, but the sequence is a little bit different, and that's the comparison that you have there. It's called mapping a gene. Mapping a gene means you have a reference genome that you're comparing against. Like, let's say that this is the one organism that I want to compare against, and figuring out how closely related everything is to this one organism. The gene itself matches up pretty well. It just has some gaps within. And so then Smikes' question is, how do we know that this gap here, there isn't just something missing? Like a mutation, like a one-off mutation. So Smikes, the way you know is you're comparing this gene, but you're doing this to everywhere in the genome at the same time. And so then that kind of washes out the effect of, yes, there might be like 1 or 2 mutations between individuals of the same species, but that percentage difference will be so small that that's not driving the difference in the phylogenetic measurements. Instead, what's driving that difference is a global difference between the organisms. So the one-off won't influence the overall metric and the statistics behind it. But when you have a lot of these events, that'll drive it. They do have the morphological data. So this is the new newt that was discovered. Again, it's the crocodile newt. This is one of the males. And it's the holotype measurements. That's what they go and compare back against. This one was alive when they took the images. Oh, yeah. Female, male, male. Female, male, male is what they have. So they have different comparisons between them. I think they're like, it's a nice set of high-res images that they've taken on these animals. And then here is the underside, so the ventral side of the animal. Very different, yeah, between the back side, between the back here, like the dorsal view and the ventral view, which is kind of interesting. Interesting that males aren't more attractive, is that common in amphibians. So I think sexual selection in amphibians is more olfactory and auditory from what I've read. And this is, I don't know how much of a blanket statement that is, but that's just what I've seen. It's not so much a different colors and not visual. And then here, I like that they have these figures in as well. We had one of these last week. These were the different collection sites. They identified existing species in the past located here. And then right here is that really high elevation in Vietnam that they marked with the star. That's the new species that they identified. So they have that one new species site, And then here is other sites where they've also discovered close relatives of the crocodile newt. And I thought, again, it was kind of cool that they're putting in the map. Not something standard, unfortunately, in all these papers, but it's something that I really enjoyed seeing. And then they have this, this is what the habitat looks like. But again, really nice papers. Here are some of the measurements that they took and had comparisons between species and between sexes. One thing that kind of I did not enjoy with these graphs that they had in terms of the measurements is the axes. If you're just quickly looking, you might suspect that there's not much difference between males and females. But if you look at the axis, this one goes from 6 to 10 and this one goes to 7 to 11. So that's just one thing that kind of threw me. I did not enjoy seeing that because it can be very quickly misleading. That's the new species was a newt. And that is your science news story of the evening.